We're going to talk tonight about the origin of life. As you know, it's been suggested that things have gradually improved, starting with some primordial protoplasm, that natural selection acted upon the varieties that were made by the mistakes in this protoplasm in order to produce all the living forms that we have. And here we are, the result of millions upon millions of mistakes in the reproduction of some primordial protoplasm. Now, that's fine, and you agree with me. Now, the only difficulty is, where did that bit of primordial protoplasm come from? Because we all know that if you were to kill a mouse, gas it, say, there's no way you can get it back again, even though it's got all its organs and everything. So, the problem is the very first form of life. It obviously needed to be very, very simple. And if you go back far enough, they say, well, it has to be just chemicals coming together. I'll quote to you from one of the great friends of creationism in the British Isles, Richard Dawkins. Well, I'm serious there, because he goes so much over the top that it rebounds to our benefit. He says, the first living cell arose, and from that we all descended, be it plants or animals or humans. That's an established fact. They're all cousins. Scientists accept it. Not to believe it would be absurd. If we go back, we find that all life forms have the same code, the same genetic code, the same metabolic pathways, the same fundamental chemicals. And so it would seem that life started only once. Whether it was evolved, as Dawkins said, or whether it was created, as this book suggests. There's only one beginning. Darwin suggested that there was some warm little pond somewhere. Alexander Ivanovich Oparin, back in the 1920s, wrote papers about this and suggested that some very simple chemicals came together, and if you give them millions of years to react, you put in lots of energy, sometime or other, just by chance, the right stuff will come together, bonds broken, bonds made, until the first self-replicating, simple life form arrived. And from that, we all descended. Not to believe this would be absurd. In the West, the idea was taken up by Professor J.B.S. Haldane, who was also, incidentally, a communism, along with Oparin. And, of course, communism's based upon atheism. And if you're an atheist, you've got to believe that somehow or other, life started by chance. The idea was that there was a primeval soup. Back in 1951, Professor Urey and one of his PhD students, Stanley Miller, did some experiments. They put together in a flask some water to boil. They had some gases going in. There was methane, hydrogen. There was ultraviolet radiation. Well, there was some ammonia as well. They found they needed that. And they passed a lot of voltages through it, a bit like Frankenstein being shaken into life by electricity. And wonder of wonders, they got some organic chemistry. Now, any first-year student at university would tell you that they were going to get some organic chemistry and they'd have some simple products. Incidentally, it was a bit of a tarry goo. They had to separate it out with a cold trap, and then they analyzed it. They found that there were a few amino acids. Now, amino acids are the building blocks of life, and therefore the headlines in the newspaper said, scientists have created life in a test tube. Now, of course, what they made was a few of the 20 amino acids that occur in nature in proteins. They made an awful tarry mess as well, which would have destroyed any enzymic properties that a protein might have had, and in any case they didn't get the proteins. They just got the building blocks for proteins. 40 years on, Stanley Miller looks back, and this is quoting from Scientific American, the problem of the origin of life has turned out to be much more difficult than I and most other people imagine. And there you have it straight from the horse's mouth. Now, the headlines in the newspapers didn't at that point say, scientists haven't made life in a test tube. They kept very quiet about that. In a recent New Scientist, I think it was about 2 weeks ago, they said, do you want to impress your mates? Talk about your knowledge of the origin of life? Well, a good place to start is this Stanley Miller experiment. Now, they said it was bluffing. And that is exactly what it is. Bluffing. Nowadays, they talk about the hot chemicals at a mid-ocean ridge. And they say that these hot chemicals, very concentrated in water, which if they were at ordinary pressures would have boiled to steam, that these soups may have had the beginnings of the origin of life. The great advantage of the mid-ocean ridge is that you don't have any oxygen down there, because oxygen would kill off all these chemicals and destroy them and burn them up. Organic chemicals don't last so long in the presence of oxygen. Now, the other place that they suggested was somewhere different, like Mars. Now, there's a good idea. You see, if you can say it's far away and long ago, like Mars, and you've got impacts coming, you've got water coming up to the surface, life developing, very simple life, then it gets knocked off, a bit of Mars comes down in the Antarctic maybe, then you look at it, and it's got what they call nanofossils. Now, nano simply means that it's very, very small, 10 to the minus 9 of a meter. Now, they made a tremendous fuss about this. They said, there's definitely fossils here, not life as we know it, but nevertheless, nanofossils. They came from Mars. They've been hanging around in the Antarctic for so many millions of years. I don't know how they knew that. They also say that it was, of course, on Mars for millions of years. And they think that they've discovered some canals on Mars where there used to be rivers anyway, and possibly below the surface there's some water. And wouldn't it be a good thing to go up and have a look? And they used this to get public opinion on their side for yet another Mars probe. In the New Scientist, for the 8th of August 2 years ago, there was a whole article about this, and it said that the meteorite nanofossils claim was deliberately hyped by NASA to obtain funding for a further Mars probe. So there's a lot of politics in this. There's a lot of economics in it as well, and people are ready to swallow it. They talk about little green men on Mars. Now, they've since discovered that these microfossils that you've been looking at are nothing of the sort. They're inorganic in origin. They certainly don't represent anything that was alive once upon a time, oh my best beloved. They're simply natural occurrences by ordinary physical processes. Since the time of Oprah, our knowledge of the cell has increased considerably. We no longer think of it as just a blob of protoplasm. The cell wall, for instance, is able to allow certain things in, such as food, and to let certain things out, such as waste products. But there are other things which it won't let out, and things which it won't let in. So it's almost as though you've got a sentry on duty on the wall. And inside the cell of eukaryotic cells, you've got the nucleus with nucleic acid, the DNA, which is a double helix. This is two strands joined together by hydrogen bonds, which are about 1/10 of the value of the power of a chemical bond, which means to say that during cell replication, the hydrogen bonds unzip themselves very easily, and each of the single strands that's formed acts as a template for the other side, so that you get two strands formed out of the one. Now, beyond the nucleus, within the cell, you've got lots of other things which are loosely called organelles. For instance, you have ribosomes, where the proteins are made. And this protein factory turns out proteins with the help of another nucleic acid, RNA. The DNA passes the information to make the protein to the RNA, and then the RNA in the ribosome, with about 100 other proteins, produces the protein that's required. So you have proteins used to make proteins. Now, another of these organelles in animals is called a mitochondrion, and that's where the energy is made. In plants, the same thing is called a chloroplast. These are absolutely essential to the well-being of the cell. Cell cannot manage without one, and yet you have something very complex as well as the rest of the cell. It's been suggested by Professor Margulis that once upon a time the mitochondrion was independent of the cell, and they acted in a sort of symbiosis. And then they got on so well that the mitochondrion was actually absorbed into the cell, just as we see it today. The cell is no use without the mitochondrion. The mitochondrion can't exist without the cell. And so that does seem a rather uncertain and unlikely picture. Also, another of these ribosomes is called lysosome, and the function of lysosome is to get rid of foreign material. And so if the mitochondrion had come in as a foreign piece of material, then the lysosome would have sorted it out. So it rather knocks that idea on the head. I want you to get the idea that the cell is a very complex business, not just a blob of protoplasm. There are two major chemicals in the cell. There are the proteins, which are a string of amino acids, and there are the nucleic acids, such as DNA, RNA, transfer RNA, messenger RNA, and so on. And we'll look at those briefly. Firstly, then, the proteins. Proteins are made up of amino acids, and I need to say just a little bit about amino acids to start with, because amino acids are the things that you can make in a Miller-type experiment. Now, amino acids have a central carbon and 4 groups around them arranged tetrahedrally. One of the group is hydrogen, another is the amine group, the NH2, a third is the carboxylic acid group, COOH, and lastly there is another organic group which we'll just call R, and which varies according to the amino acid. As I say, there are 20 different amino acids found in natural proteins, living things. There are lots of others that can be made and have been made using the Miller experiment, but they're not found in living things. Now, the great thing about this is that because they lack a center of symmetry, you get 2 forms with the same formula. They're known as stereoisomers, and they're a bit like your hands. You've got a left hand and a right hand, and there's no way in which you can superimpose one upon the other. There isn't a way of doing it. They're different, but they're mirror images of one another. If you had a mixture of amino acids, D forms, dextrorotatory, named after the way in which they rotate polarized light, and L forms, livorotatory, they're sometimes called R and S, rectum and sinister or something. But if you've got all sorts mixed together in a chain, then they would lose their 3-dimensional shape. If you have them all left-handed, as always occurs in proteins, Then you get a spiral shape, which then collapses into a three-dimensional shape, which has grooves that can grab hold of things. For instance, the spittle that you have in your mouth has a particular enzyme which is shaped a bit like your fist and can grab hold of things. And there are active in groups, chemically active groups inside that, in exactly the right place, which allows it to do a specific job. Some enzymes do have a specific job. Others can do lots of things. For instance, the digestive enzymes that are all working in us at the moment, they can cope with everything from potatoes to beef and turkey. Now, it's difficult to see how you would finish up in a natural experiment with just the L form of these amino acids. Now, on the law of parity, you should get a 50-50 D and L form. That's a racemic mixture, as chemists call it. In the Miller experiments, you got racemic mixtures only. As I say, difficult to imagine from a theoretical point of view how it could be anything else. And yet, in a New Scientist very recently, I've got a copy of it here, it talks about the possibility of getting just L forms. That's this one here. Now, what it's saying is that polarized light, in the presence of polarized light, one form will decompose ever so slightly faster than another form. Now, the big problem about that is that you don't find a lot of polarized light, except on one side of a pair of sunglasses. So they then said, yes, but there are some metal oxalates, I think it is, that they mention, and they are also stereoisomers, and one form will decompose ever so slightly faster in the presence of ordinary light, provided that there's a strong magnetic field. Now, where do you find a strong magnetic field like that? And because it's going to happen with these oxalates, is it going to happen with the D and L forms of amino acids? Also, the difference between the decomposition rates is 1 part in 1,000. So they say, well, over millions of years, maybe you finish up with just one form. But of course, you've also got, fighting against that, a racemization process anyway. In fact, they use that as a dating method. They got this scale completely wrong, of course, as usual with dating methods, but they do use this racemization method. And so, again, they're clutching at straws. Now, why I'm saying this to you is that if they're making such preposterous suggestions as this, and clutching at straws, it shows that they really have no idea as to how they could get L amino acids, 100% L amino acids. Do you know that you have within your liver enzymes which specifically seek out and destroy any D amino acids? Because they're poisonous. They would ruin any protein that your body was going to make. So, I point that out because if creationists were to clutch at straws in that way, the scientific community would be very fast to say, look, they haven't got a leg to stand on. But yet, because they're so desperate to prove that not to believe this would be absurd, as Richard Dawkins says, that they come up with these ideas. It's amazing, isn't it? Now, the amino acids, how do they join together? Well, 2 of them come together, they lose water. There's 2 amino acids, the COOH and the NH2 come together, the elements of water, H2O in the middle, come away, and then they join together by what's called a peptide link, and that's what's then known as a dipeptide. That continues until you've got a whole chain of them. But any old chain wouldn't do. They have to be in a particular order, so that you've got a glycine there, an alanine there, and so on and so forth. 20 different ones, but not in the higgledy-piggledy order, a specific order, so that when they then, the whole thing loses a coating of water and it condenses into a particular three-dimensional shape, then it's ready to do the job it's designed for. And I really do mean that, it's designed. It couldn't be something that occurred by chance and natural selection. Remember that until the first cell was living and reproducing, natural selection cannot come into the picture. Consequently, you can't claim natural selection as being an ally in producing something bigger and better. So, to do this, you need the DNA with its information, which then passes it on to the RNA. The RNA and something like 100 or more different proteins come together to produce just one of these long chains. It's a very complex business. Each part of it is carefully designed. If you didn't have one little bit of it, it wouldn't work. And if proteins are used to make proteins, there is a problem. How'd you get the first protein? If we look at the Miller experiment again, we'll see that at the end of it, because you're putting in so much energy in the form of ultraviolet radiation and the spark that you see at the top there, between those electrodes, then it's going to produce lots of decomposition. And you finish up with a tarry mess. And yet, in the living cell, you get 100% production of the very thing that you want. It's pure, and there is no tarry mess. You see, if you did have a tarry mess, it would have active groups in it, which would then react with the stuff you wanted to produce and turn it into something else and decompose it. And it would certainly lose its enzymic properties in the presence of a tarry mess. When God does things, He does it really well, I have to say. He's not just a mathematician of a high order, as I think it was James Jeans said, but speaking myself as a chemist, he was a biochemist of a very high order as well. These molecules are destroyed by the very conditions that make them. And that's why you've got the condenser there and the cold trap at the bottom to take them out of the conditions. Because if you left them like that for millions of years, they'd simply decompose. I'm just talking in the terms that everybody else uses for this. Now, these very often are reversible reactions. A reversible reaction is one where you've got a forward reaction of products going to, reactants going to products. You've got a backward reaction of products going back to reactants. They're in a dynamic equilibrium. When it reaches final equilibrium, it means that the rate of the forward reaction equals the rate of the backward reaction. There's no overall change. It's actually changing all the time. It's a dynamic equilibrium, but you reach a point where you're not going to get a lot of difference in the product. Now, in most of these cases, in the equilibrium composition, there are more starting materials than there are products. So if you leave it, it just ensures that it reaches equilibrium. And equilibrium is in favor of the starting materials rather than the products. So time is not an ally in this situation, because you're not involving any form of natural selection or any sort of selection. So you can't say, oh, given millions of years, this could happen. It just won't. These proteins are thermodynamically unstable. That means to say that they prefer to decompose, certainly in the presence of energy, such as heat, ultraviolet light, electrical impulses, and so on. They're also unstable with respect to water. because you've seen that when you bring them together, you're actually eliminating water. So if you were to make, should we call it a proteinoid, a homemade protein, chemical protein, then it would tend to decompose in the presence of this soup, this watery soup. It's unstable then to products of the reaction as well. In other words, it's thoroughly unstable. It's very difficult to decide how this could happen. When they do these experiments, Miller and those that have followed him and jumped on his bandwagon and tried no end of different, so many laboratories have tried to do these experiments and to produce different things which they think might be biologically significant. They always start out with 100% pure starting materials. But if they're trying to reproduce what happened in a prebiotic earth, and of course there wasn't a prebiotic earth except for the first day or two, but if they were trying to reproduce what happened in that situation, you wouldn't start off with 20 grams of pure whatever, lysine or whatever. So it doesn't represent what actually could have happened even. Also, they didn't have any oxygen in this experiment. Now the reason for that is that the organic materials burn up in the presence of oxygen. They're decomposed by oxygen. If you haven't got any oxygen, then you don't have any ozone. Ozone is a form of oxygen with 3 atoms instead of 2 atoms together in the molecule. If you don't have any ozone in the layer in the top of the atmosphere, then you don't get any protection from the ultraviolet radiation from our sun. We were talking on the news this morning about the way in which this ozone layer has been dissipated and people are finding water instead of ice at the North Pole. Now, if there was no oxygen, there'd be nothing to prevent ultraviolet radiation from reaching these chemicals, and in particular the nucleic acids are vulnerable to ultraviolet decomposition. They break down very quickly, much more quickly than they could possibly be made. So the absence of oxygen doesn't really help at all. If you don't have oxygen, the nucleic acids are decomposed. If you have oxygen, the proteins are decomposed. You can't have it both ways. Miller's experiment is founded upon hope rather than science. Now, we've looked at proteins, and we must just quickly, we're okay. We'll look at the DNA and the RNA, the nucleic acids, and you'll see on the right of the diagram that the DNA is in the form of a double helix. And we achieve this configuration because all of the sugars are right-handed. Now, just as we saw that amino acids were all left-handed in biological systems, so the sugars are all right-handed. They've been selected, and nobody knows quite how this happens. My own theory is that God made the proteins with his left hand while he was making the nucleic acids with his right hand. And then on the left of the diagram, you see the chemistry, those of you who are in the front row. And you've got a sugar phosphate background. And the sugars are joined through the phosphates from 3' to 5' on the sugars. And it's always like that. And that's not the thermodynamically most stable form. It's not the form you'd get if you were doing this in the laboratory, but it's formed like that in order to be biologically active, and it's done using proteins as templates. So trying to do it by chance just doesn't work. Now, on that backbone of sugar and phosphate, you've got 4 different bases. Now, any one base will only pair with a particular other base. And so when these bases, which are connected by hydrogen bonds, which are very weak, when they break and the whole thing unzips, then one side acts as a template for the other side when the cell splits and replicates itself. The DNA carries information. It was at the Second European Creationist Congress that Professor Werner Geertz of Germany pointed out to us that as a carrier of information, the DNA molecule is 45 million times more efficient than the man-made silicon megachip. Now, imagine if it was 45 times as efficient as your computer's megachip. And we're talking about megachip now, a million bits of information on a silicon device. If it was 45 times as great, my word, that would represent a tremendous advance, technologically speaking. It would certainly sweep the market and replace everything else. It would be like replacing steam. Now imagine, multiply that by 1 million, and then as though that's not enough, multiply that by 1 million, until you got 4.5 times 10 to the power 13 times. Now, we all know that the man-made megachip is the result of teams of people designing, calculating, working their socks off. None of it arrived by chance, and you'd certainly upset anybody who was working on it by telling them, well, if you'd waited a few million years, that would have turned up anyway. All you need is to extract the silicon from the silicon oxide of the sand, and you're home almost. Well, if we say that the megachip had to be designed, what about something which is 4.5 times 10 to the power 13 times more efficient than the megachip? Did that really come by chance? Not to believe this would be absurd, says Richard Dawkins. Who is really being absurd here? Well, there are 4 of these bases, and any 3 together in juxtaposition to one another acts as a codon and is read by the RNA, and it is used then to make a particular amino acid, so that you've got one particular codon will start a sequence, another codon of 3 such bases will finish a sequence, and in between you've got a whole string of them that produce a string of amino acids in exactly the right order to produce a protein which has particular biological activity and significance and purpose. Now, there are 64 such codons, of course, because you've already worked out that 4 times 4 times 4 equals 64. So these are, there is information involved, there is purpose, there's design. And we understand from information theory that you don't get information by chance changes. You actually lose information by chance changes. In the biological field, chance changes are called mutations. We also know that information can only come from an intelligent source. So genetic information, let's say the information to make our cat at home, it's a real stupid moggy. It's not his intelligence that's produced this DNA, but it's the one that made the cat in the beginning. Information only comes from an intelligent source. This shouts creation. And incidentally, if mutations lose information, then neo-Darwinism, which says that evolution takes place by virtue of a number of mutations, some of which might be beneficial, although as yet we've never found a mutation which adds information, then that is impossible. It goes against the tenets of information theory. Neo-Darwinism is not a possibility for a mechanism of evolution, biological evolution. To believe this would be absurd. In a living cell, then, the proteins are coded for by the DNA. The DNA itself is made using proteins. It's constructed by use of proteins to get the position across the sugar right for the phosphates joining them. Now, if the proteins make the DNA codes for the proteins, one has to say which came first. It's a chicken and egg situation, isn't it? They had to all be there together. Now, when it comes to talking about these things, you'll find that the literature follows Darwin's ideas. Darwin said, it may be, it is possible that, probable that. Now here is a picture of a page discussing the RNA as the origin of life from a standard biological textbook, and you can see that it's full of imagines. May have been, presumably, nearly, perhaps, and so on. And it goes on like that for the whole of the page. This is not science, and certainly not a basis to stand up and say, well, this is definitely a fact, and not to believe it would be absurd. Incidentally, not only does Miller not produce any RNA or DNA or proteins, but proteins can't reproduce themselves. RNA doesn't reproduce itself. And so you're stuck with looking for something which might have made the first cell. It all had to be there at the beginning, and each component is breathtakingly complex. In isolation, it couldn't survive. It couldn't replicate itself in isolation. To do the replication, you need the whole lot acting in concert. All the parts of a cell are needed to function and to replicate. Nothing works unless everything works. Now that's what Michael Behe in his book Darwin's Black Box calls irreducible complexity. Now it's not just the whole cell which is irreducibly complex, but components of the cell are also irreducibly complex. And I want to show you one particular component of the mitochondrion, which you'll all remember is the bit of the cell where energy is made. Now, within the wall of the mitochondrion, you have little motors, and you have hundreds of these in every mitochondrion of every cell. These motors are so small that you could fit 200,000 of them into the space occupied by a pinhead. So we're talking micro technology here. And there's a picture of it. The cell wall of the mitochondrion has protons going up through, and as it passes through this, so the wheel of this enzyme, which is known as adenosine triphosphate synthase, it starts to rotate. And it rotates at 100 revolutions per second. And that's just when it's ticking over. It can go much faster. And at each rotation, 3 sets of adenosine diphosphate plus phosphate is converted into adenosine triphosphate, which stores the energy of the bond to phosphate. And that ATP, as it's called, adenosine triphosphate, can then couple itself to a reaction somewhere else within the cell in order to do work and use that energy, and then it breaks down into adenosine diphosphate and phosphate again. So the whole thing is recycled. And it wasn't just your local authority which invented recycling of waste products. This has been going on since the very first cell. And these things are found in every plant, in every animal, in every fungus, in every bacterium, in every form of life you've got this most complex enzyme. Now, when you're just sitting around, because your brain's in gear, your heart's beating and so on, you recycle your own body weight of ATP every day. And if you're doing really hard work, you can recycle 1 ton of ATP. And because you don't have a 1 ton in your body, it means that everything's recycling. You'll see that the central spindle, which is white in this diagram, is squeezing the 6 blobs above it. And 3 of them are opening and closing as the spindle goes round. And ADP and P are going in, and ATP is coming out. And it's all driven by protons. Now, this little motor has 31 separate proteins. And each of those proteins will have maybe 1,000 amino acids. And it's in a precise order. If any one of those proteins was missing, this motor wouldn't work. Nothing works unless everything works. It's irreducibly complex. And if the amino acids were in the wrong order in any of these, it wouldn't work. So it had to be right from the beginning. And you know, just as a plant for manufacturing cars is much more complicated than the car, it turns out, so the proteins and the DNA, RNA that would be used to set up this adenosine triphosphate synthase, that has to be much more complicated than even this motor itself. Now, on Thursday night, Dr. Stuart Burgess is going to be talking about this concept of design. He's a design engineer himself at Bristol University, and he's written a book, Hallmarks of Design, which we hope we can offer you copies of. I'm really looking forward to hearing Stuart. I want to just close by pointing out this. When the Antarctic meteorite was found, and they said, oh, that's got nanofossils in, they were hoping that there was life on Mars. They're not so sure now that there's life on Mars. They're desperately looking for other planets. They say that they found several other planets, but not in our solar system. All the planets in our own solar system are incapable of supporting life, except one, the Earth. And it says in the book I was reading that the Earth is made to be inhabited. It does seem like it when you compare it with Venus and Mars and Jupiter and so on. And even bits of the moons of Jupiter, like Io, that they're desperately looking for water on. And they say if there's water, then pretty certainly there's life. Not to believe it would be absurd. And at the moment, all of the planets that they've found are not actually because they've looked at it and said, oh, look, there's a planet. What they've seen is a wobble in a star. Now, sometimes the wobbles in the star turn out to be wobbles in the telescope. They found one which had a period of 365 and a quarter days. And then they thought, well, that's a coincidence. And then they realized that they hadn't taken account of the Earth's movement itself. Well, it's possible that some of these wobbles do represent a massive star, but it has to be a massive star for there to be any sort of wobble in this, a massive planet, if there's to be a wobble in the star. And these massive planets, like our own solar system, Jupiter, for instance, they're incapable of supporting life, even if life could arise in the way that Professor Dawkins seemed to think. The sheer gravity, for one thing, would make it very difficult. You'd need to have exactly the right conditions. And when you look at it, the Earth has got exactly the right conditions. In fact, people have pointed out that the whole universe is almost as though it was designed for man. And they use a Greek word, and they say it's the anthropic. Anthropic is all Greek to me. The anthropic principle. And then people say, hang on a minute, that sounds a little bit religious. Maybe the real answer is that we're just one of millions of universes. And because one of them had to get it right, we're on the one that's right. If we weren't on that one, we wouldn't know anything about it anyway. So there's a possibility, and someone suggested it's a bit like the suds of soap that you get when you put fairy liquid in the washing up dish and stir it round. And each one of those represents a separate universe, but they're all connected, and maybe there are black holes which have wormholes connecting them, one universe to another, and you could go down one and finish up in a completely different universe. And in support of that, I would say, well, where is Lord Lucan? Anyway, that's just the thought. If we're desperately looking for ET, extraterrestrials, to help us make our telephone calls and so on, then this is the basis for a great deal of what passes for entertainment on television. There's a whole industry, books galore, all written about it, Star Wars and so on, to boldly go. And what they're looking for with the SETI program, the Search for Extraterrestrial Intelligence, is a non-random radio signal, because if there's people out there which are as intelligent as we are, then maybe they're going to try and communicate with us. Well, if they could only find a non-random signal. They thought in Australia a couple of years back they'd found it, and then they realized it was about the time that everybody was coming home from work and opening up their garages with their remote controls. Now, we're looking for a non-random sequence which has meaning and purpose. And then they look at the DNA, they see a non-random sequence of nucleotides which has meaning and purpose, and they say, that arose by chance. Not to believe that would be absurd. Thank you.